talk is titled Unraveling the Molecular Mechanism of Interactions of the raw GTPases CD42 and RAC1 with the scaffolding protein IQGAP2. Just before that, the next speaker, please come forward in a while in a quiet manner to get your microphone set up.
Okay. Hello everyone, I'm Sila Ozdemir. I got my PhD degree last year from Koç University in Turkey, and currently I'm a postdoctoral fellow in Oregon Health and Science University in the United States. This work that I'm going to present today is belong to a part of my PhD thesis, and during that work I collaborate with Ruth Nisinov group from NCI in Fort Detrick. And the talk will be about the interaction mechanism between raw GTPases and a scaffolding protein called IQGAP. This work has been published last year in Journal of Biological Chemistry, and also it has been selected to 2018 collection of JBC as the representative of the computational biology field. So first, I'm going to talk about the main concept from the project. So raw GTPases, similar to other GTPases, they hydrolyze GTP to GDP, and they comprise over 20 human members. These raw GTPases are the subfamily of RAS superfamily GTPases, and their main functions is to regulate actin polymerization, cell growth, and cell motility. And as well as other GTPases, raw GTPases also have a binary switch. It means that they are in their active states when their GTP bound form. However, when this GTP hydrolyzes back to the GDP, they are inactive. And upon this activation after binding of GTP, there are some conformational changes occurring on the switch 1 and switch 2 regions, and these conformational changes enable to binding of several effector molecule to the GTPase molecule. And raw GTPases can be distinguished from the other GTPases by the presence of the insert loop. This is the insert loop, and it's an alpha helical motif that's enriched by the charged residues. RAC1, CDC42, and RAW-A are the most studied raw GTPases. Today I'm going to focus on CDC42 and RAC1. These two proteins are the key regulators of actin reorganization. They are playing a role in actin polymerization and cell motility. So even though these two proteins has a high sequence identity between themselves, their function, the role they play in actin polymerization are different. So when I look at the sequence of these two proteins, I realized that, and you can see that in their important regions, such as switch 1, switch 2, and insert loop, there is a striking sequence differences between these two proteins in their insert loop, and most of these amino acid differences are charge reversal differences, actually. So CDC42 regulates actin polymerization by interacting with Wasp protein, but however, RAC1 doesn't involve in that pathway. It has its own way to regulate actin polymerization. So their function and the mechanism of acting on the actin polymerization different than each other. And IQGAPs are the other protein family that I'm going to focus today. They have 3 isoforms, IQGAP 1, 2, and 3. IQGAP1 is ubiquitously expressed in almost every eukaryotic cell. IQGAP is a more tissue-specific protein. It's found predominantly in liver. It's reported that all 3 isoforms of IQGAP can interact with CDC42 and RAC1 in their active form, in their GTP bound form. And it's known that CDC42 is stabilized in its active form upon binding to IQGAP. So when it's bind to the IQGAP, it's always stable, active, and in its GTP bound form. And also it's known that CDC42 and IQGAP bind to Wasp protein, and they can synergetically activate actin polymerization and cell motility. It's known that in some cancer types, IQ-gap protein is overexpressed. So it means that when this protein overexpressed, it also increases the rate of active CDC42. So the overexpression also increases cell motility by activating actin polymerization, and it can cause metastasis as well as following the cancer. So it's important to know the details of this interaction because their role is shown in the metastasis. So IQ-gap is a large protein, huge protein, so it has some different subdomains responsible for the binding of different effectors. Actually, it interacts with over 100 different proteins. And GRD domain is the domain that's responsible for CDC42 and ROCK1 binding. 2 or 3 years ago, the crystal structure of CDC42 and GRD complex has been revealed, and here it's shown that there are 2 different binding sites on GRD for CDC42. They are called EX domain and RAS capsid. Actually, EX domain in the 3D structure came together and created an interface like this blue part, and RAS capsid can be seen here. So in the crystal structure, there's GRD and 2 CDC42 bound this GRD from EX domain and RAS capsid. And interestingly, in the crystal structure, it has been revealed that GRD is actually dimerized. So in the whole structure, there are totally 2 GRD, which is in dimerized form, and there are totally 4 CDC42. However, the exact binding mechanism of CDC42 to GRD is not known. So as I said, this interaction is important because they have role in metastasis. So exact binding mode and mechanism should be revealed. So one of the main questions that I tried to find answer in this project was the exact binding mechanism of CDC42 to GRD. And this is the side view of this protein without any CDC42. So you can see that this 2 GRD dimerize, and their C terms play an important role in that dimerization. Actually, this C term belongs to that protein, and this C term belongs to this one. Their C term actually tangled with each other. The interface for this interaction, actually the C term of this 2 protein. However, the role of CDC42 in that dimerization also is not known. It's the second question that I'm going to seek an answer for it. And that's the situation for CDC42 and GRD binding, but binding mode or binding stoichiometry for ROCK1 and between ROCK1 and IQ gap is not known. So I'm going to try to answer what's the binding mode of ROCK1 to IQ gap, and I'm going to try to answer whether this will be the reason their different mechanism in actin polymerization. So for this purpose, I simulate that 5 complexes using NAMD platform. So this 3 structure taken from the crystal structure. This is the free GRD, and this is the GRD with EX mode CDC42. And this GRD has also CDC42 bind to its RAS capsid. These 2 models, actually there is no crystal structure revealing the ROCK1 and GRD interactions, so I model these complexes using PRISM web server. I predict the interaction and complex between ROCK1 and GRD2. So I simulate that 5 complexes for 500 nanosecond long. So after analyzing the simulation, I realized that when I compared free form of GRD with the one that have a CDC42 in its EX domain, the C term of here is the C term of these 2 proteins differently behave. I will show it again. In that case, the C term is folded upon itself. However, after CDC binding, you can see that C term is released. And it means that since I show you that C-term is the platform interface that playing role in dimerization of GRD, so upon binding of CDC42, C-term is released, so it may imply that CDC binding to EX domain may facilitate GRD dimerization. Also, when I compared the RMSF plots of these specific domains, this is the blue part, you can see that fluctuation of these particular residues is much more higher compared to free GRD2. Then I closely look at the binding mechanism between CDC42 and GRD2 from EX domain, and I saw that insert loop, the loop specific to the rho GTPases play an important role in that binding. So when I closely look at the salt bridges and hydrogen bonds stable throughout the simulation, I saw that there are lots of stable bond between insert loop and GRD2. So I conclude that CDC42 binds to GRD, EX domain of GRD through its insert loop. Then I compared EX mode binding of the RAC1 to CDC42 binding. It's known that CDC42 can stably bind that side, but we don't know about RAC1. So when I look at, this is the average conformation of the simulation. When I look at the insert loop of RAC1, you can see that orientation is quite different than the previous one. And I couldn't observe any stable hydrogen or salt bridge between these two parts. So insert loop is different than the CDC42. And when I compared the binding free energy, binding affinity between RAC1 and EX mode, it's quite, let's say, smaller than the affinity between CDC42 and GRD2. And when I compare the sequence of this insert loop of CDC42 and RAC1, you can see that there are like charge reversal differences between these two regions. So these differences between insert loop of CDC42 and RAC1 inhibit its binding to EX mode of CDC GRD2. And when I look at the movement of C-term and whether it's released or not, it behaves similar to the free GRD actually. So RAC1 binding to EX domain doesn't initiate a C-term release. So it means that only CDC42 binding to EX domain can initiate a C-term release so far. Then I look at the RAS-CAP site binding of these two proteins, CDC42 and RAC1, and their binding mode quite different than the binding mode of CDC42 to EX domain. The primary binding site of CDC42 and RAC1 in that case, switch 1 and switch 2 region. Insert loop doesn't play an important role in that case, and their binding mode is quite similar. And then when I look at the C-term release after binding of RAC1 or CDC42 to RAS-CAP site, I couldn't observe any C-term release. So I can conclude that only CDC42 binding to EX domain can induce that C-term release, which eventually facilitate the GRD dimerization. So overall, I propose that model. First, CDC42 come and bind to EX domain of GRD2 using its insert loop, and it releases C-term of the GRD2, and it also facilitates some allosterical changes on the RAS-CAP side of the GRD2, and it's enabled the binding of second CDC42 to that side, and this binding is facilitated by switch 1 and switch 2 region of CDC42, and overall this complex can dimerize because its C-term is in its free form, its release. However, in the case of RAC1, it cannot bind to EX domain because of the sequence differences in its insert loop. However, RAC1 only can bind to RAS-CAP site, so this stoichiometry is 1-to-1, even though it's 2-to-1 in CDC42. So only one ROC1 can bind to GRD2, and it cannot induce any release in the C term. So it's the overall complex for the ROC1 and IQ gap interaction. So it's important that maybe this different stoichiometry between these two proteins, important to explain the different mechanism, different effect mechanism of on their actin polymerization. And as a future perspective, it's worth to investigate the like interaction between IQ gap, CDC42, and VAS protein, because as I said, they are synergetically act on VAS protein, and they initiate actin polymerization. So it's important to look at that interaction of that complex too. And also interaction of GRD with other raw GTPases can be also investigated. I would like to thank you, my PhD advisors, Özlem Keskin and Atilla Gürsoy. Also, I would like to thank Ruth Nisunov, Huyumbam Zheng, David Sachs, Zigang Li from NCI, and also I would like to thank you, ISCB and 3DSIC for their financial support and also for the opportunity. And as I said, I'm a postdoctoral researcher right now in Oregon Health and Science University, and I'm working in the Cancer Early Detection Advanced Research Center. So we are focusing on the developing early detection methods for the cancer. So we are recruiting new people from different backgrounds. So if you are interested in, you can come and go and check this website, and also you can come and talk to me if you are interested. Thank you very much for your attention. I would like to accept the questions.
Any questions for Emine?
Hello, thank you for your presentation.
Thank you.
I was just curious. I just saw there was a metal around your active site. That's magnesium ion. Were you able to calculate for any metal parameters before setting up your molecular dynamic simulations?
You mean the models of the...
In one of your proteins, I guess. Mm-hmm. Yeah, you have a metal ion that is magnesium, I think, around your active site. Were you able to calculate for any metal parameters before setting up MD?
Actually, like in my topology and parameter file, I also included that magnesium ion. I didn't just exclude it. So it's in the simulation, and it's also calculated via calculate binding pair energy and the other analysis like fluctuation profile. So its contribution, the ion's contribution, is also there.
Oh. Which force field was that you used to calculate for the parameters during MD?
I used charm.
Okay, thank you.
Thank you very much, Emine.
Thank you.
Your name reminds me of the Emine.
Actually, it's Emine, but okay.
I apologize.
No problem.
So our next speaker will be Jan Kozinski, who will speak about